So they are able to tolerate high temperature, they are able to tolerate very intense, bright, 100% full days, full day type of noon type of summer sunlight. They are able to tolerate saline condition, they are able to fix carbon dioxide with a double amount or double faster rate, and then they transpire less, so they save water. That's why these plants are better compared to the C3 plants. And another important point is that they continue to perform photosynthesis even when their stomata is closed. So whenever stomata are closed, remember C3 plants will stop photosynthesis, but C4 plants have the ability to maintain carbon dioxide concentration in the bundle sheath cell for a longer time. So they are able to perform photosynthesis even when their stomata are closed. Another important point, they have little loss of photosynthetic activity because C2 cycle or photorespiration, a wasteful process, is absent in their case. So these are the important points how C4 plants are better when compared with the C3 plants. Now let's quickly understand the differences between the C3 plants and C4 plants, so that then we can move over to the next topic. So some of the points I have mentioned here, let's start. So the first point is, in C3 plants there is only one time carbon dioxide assimilation, because there is only mesophyll cells, though mesophyll cells are of two types, but they all are mesophyll cells. So there is no difference in their functioning mechanism of photosynthesis. So that's why there is only one carbon dioxide assimilation, and that is only C3 cycle. But in the case of C4 plants, carbon dioxide assimilation occurs two times. First, it occurs in the bundle sheath, in the mesophyll cell, in the form of a 4-carbon compound, and then it is transported into the bundle sheath cell, where carbon dioxide is again liberated, and then this carbon dioxide enters into C3 cycle. So it occurs two times in the case of the C4 plants. Now in the C3 plants, the carbon dioxide acceptor is RUBP, there is only one acceptor. But in the case of the C4 plants, two times carbon dioxide fixation occurs. That means there are two times acceptors of carbon dioxide. So in the C4 cycle, PEP is the carbon dioxide acceptor, and in the C3 cycle, just like this one, there is RUBP present here. Now the stable compound formed in the C3 plant is always a 3-carbon compound, that is phosphoglyceric acid or 3-phosphoglyceric acid. In the case of the C4 plants, two times compounds are formed, stable compounds. In the first fixation, it is a C4 compound, that is oxaloacetic acid. And in the second fixation, because it's a C3 cycle, so PGA is formed. Now PGA is reduced in the same chloroplast. Remember, PGA changes into PGAL. So PGA changes into PGAL in the same chloroplast. But in the case of the C4 plants, PGA will be shifted from bundle sheath cell, and then it goes to the mesophyll cell, where it is reduced into PGAL. If you remember this process, which I told you is very crucial, that PGA will change into PGAL, but this change will not occur in bundle sheath cell but occur in the mesophyll cell. Then it comes back into the bundle sheath cell. So this reduction occurs in the same cell in C3 plants. This reduction occurs in the different cell. Now one carbon to be fixed requires 3 ATP and 2 NADPH in the C3 plants. But in the case of the C4 plants, one carbon dioxide fixation requires 5 ATP and 2 NADPH. So there is extra 2 ATPs required for every fixation of carbon dioxide. There is monomorphic type of chloroplast in the C3 plants. There is dimorphic type of chloroplast, we have already covered. Then Kranz anatomy is absent in the case of the C3 plants. Kranz anatomy is present in the case of the C4 plants. Kranz anatomy means having a halo type of structure. The reason is that there are two types of photosynthetic cell: mesophyll cells, which are tightly packed, bundle sheath cells again tightly packed, and then they are connected with the help of plasmodesmata. But the main point in Kranz anatomy is that the vascular bundles are covered with photosynthetically active bundle sheath cells, and they are present in concentric manner around the vascular bundles. That's why it is called as Kranz anatomy. Kranz stands for halo, or it stands for concentric rings in German language. Now the next is the bundle. In the case of the C4 plants, bundle sheath cells do not carry chlorophyll, but in the case of the C4 plants, the bundle sheaths carry chlorophyll. That's why they are also called as chlorenchymatous. Parenchyma which carries chlorophyll is called as chlorenchyma according to your 9th class syllabus. Then the bundle sheath cells are differentiated in the the mesophyll cells are differentiated in the case of the C3 plants. Differentiation is palisade parenchyma or palisade chlorenchyma and spongy chlorenchyma. Palisade towards the upper surface, upper epidermis, spongy towards the lower epidermis. Palisade will have tightly packed columnar cells, and in the case of the spongy cells there will be oval cells or overall spherical cells with lots of space in between. And why space is there? Because they are in contact with the stomata. So mesophyll cells are differentiated in C3 plants, but mesophils are not differentiated in the case of the C4 plants. Now C2 cycle we'll be learning next is present in the C3 plants and it is absent in the C4 plant. C2 cycle is also called as photorespiration. Now in C3 plants only C3 cycle occurs, but in the C4 plants both C4 as well as C3 cycle occurs. Now the optimum temperature for C3 plants is comparatively cooler temperature, 20 to 25 degree, to perform photosynthesis, and C4 plants, they want a higher temperature of 30 to 45 degrees to perform their best in photosynthesis. That's why they are better when it comes to global warming. Then the light saturation, these plants never undergo saturation. They can go for any amount of intense sunlight, but these plants feel saturated by the time 70% of summer sunlight is available to them. So they cannot bear too much of long sunlight duration and hot temperature. Carbon dioxide concentration best for them is